Welcome, Dr. Frank.
Hello. Okay.
So we'll start promptly at 9 o'clock. I have a brief introduction, and then we'll get underway. I guess it's 12 o'clock your time. Sorry. Okay, so I'm showing 9 o'clock here on the Pacific Coast. So I guess we can get underway. Hello, everyone, and welcome. My name is Jake Schrader. I'm the president of WSU's American Society of Agricultural and Biological Engineers, the student executive branch. Today, we're so excited to have Nobel laureate Dr. Joachim Frank join us for a seminar on single particle cryo-electron microscopy, a visualization of biological molecules in their native states. Dr. Frank is a professor of the Department of Biochemistry and Molecular Biophysics and the Department of Biological Sciences at Columbia University. He received his PhD from the Technical University in Munich, and in 1975 joined the Wadsworth Center in Albany as a senior research scientist. In 1985, he joined the faculty of the Department of Biomedical Sciences of SUNY Albany. In 2008, he moved to New York to assume his current positions. Dr. Frank's lab has developed techniques of single particle reconstruction of biological macromolecules, specializing in mathematical and computational approaches. He has applied these techniques of visualization to explore the structure and dynamics of the ribosome during the process of protein synthesis. Dr. Frank is a member of the National Academy of Sciences and the American Academy of Microbiology. He is also a fellow of the American Academy of Arts and Sciences and of the American Association for the Advancement of Science. In 2014, he was honored with the Franklin Medal for Life Science. In 2017, he shared the Wiley Prize in Biomedical Sciences with Richard Henderson and Aaron Van Heel. He was awarded the 2017 Nobel Prize in Chemistry together with Jacques Dubochet and Richard Henderson. Again, we are glad that all of you can join us today. We are also streaming live on YouTube in case we reach Zoom's max capacity, and we will hold a 15 to 20 minute Q&A session at the end of the seminar, so please hold your questions until then. And without further ado, I will hand it over to Dr. Frank. Enjoy the presentation, everyone.
Okay, thanks very much for the invitation and for your interest. I'll just get myself started here. Okay, so I'm going to talk about single particle cryo-EM, visualization of biological molecules in the native states. So to begin with, what is life? Well, life exists in so many different varieties on Earth, and it is a miracle that we behold every day. And the big surprise was that life is very, very similar as we go into the molecular details. So we can make many discoveries as we zoom in on the parts of an organism with increasing magnification. There's an organism and an organ, a tissue, cell, organelle, and molecule. So each time we get different kinds of information, and that's by definition the definition of complexity, that we cannot expect, we cannot anticipate what's going to happen on the next level of magnification. So here is a sort of a schematic of an animal cell. And you see that it's closely packed with different organelles, and which are themselves composed in very difficult, very complicated ways. And in between the membranes, and the whole thing is just very compact. And we can attempt to look at these structures in the cell itself, but it's very difficult because everything is very much clogged together. And that's why we pursue what is called reductionism. We study a subsystem in isolation in vitro, and we hope that it approximates the processes in the environment of the cell. Molecules are actually not just swimming around at random, but they are organized in molecular machines. Many molecules act in concert in a processive way, which means there is a before and an after, and time progresses in a particular way. And this all is made possible by expenditure of energy, which essentially define the arrow of time. So all in all, in structural biology, we wish to know the structures of all components, but also the way they interact dynamically. And here are a few examples of molecular machines. ATP synthase makes ATP, proteasome recycles proteins, RNA polymerase copies DNA into mRNA. Ribosome, this is my favorite molecule because it's been serving as a sort of a demo for the development of the techniques. It makes proteins. The chaperone folds proteins, dynein transports molecules, spliceosome edits mRNA, flagellar motor, and so on. Hundreds of molecular machines, very highly organized. We wish to understand in detail how they work. So there has been a technique called X-ray crystallography. Probably everybody is familiar with it, has been there for decades. And in a crystal, many copies of the molecule are arranged in regular order. Upon exposure to the X-ray beam, we get a diffraction pattern. And by ingenious methods of phasing, we get a structure out of this. And here we show Max Perutz and John Kendrew, who received the Nobel Prize in 1962 for determining the structure of hemoglobin. They were pioneers in protein crystallography. Now, for this to work, the X-ray beam must be very high intensity. The crystal must be almost perfect. And to date, more than 140,000 structures have been solved by X-ray crystallography, and they're all available in public data banks. So why do we need any other technique for structural determination? Well, because of the crystal packing, the molecules are not visualized in all conformations and binding states that are important for the living function. And what is worse, many molecules do not form highly ordered crystals, so they are essentially elusive to X-ray crystallography. The sample quantity can be a big issue as well. So there's an alternative technique, which is by using electrons. What is an electron microscope? Well, it has the same optical elements as as a light microscope. It has a lens instead of a glass lens, it has a magnetic lens, but the optical path and the principle of image formation is very much the same. Now, the very big difference is that the glass lens in light microscopy, we're limited to the wavelength of light, which is 400 nanometers. Now, there are some super microscopy that reaches better resolution, but it never gets into the range of molecules. In the range of molecules, we have to get we get in the range of the atom. So we have to get a resolution that is better than 0.3 nanometers. And in fact, electrons, when they are accelerated to 100 to 300 kilovolts, have a wavelength which is even much smaller than 1 angstrom or 0.1 nanometers. So with electrons, we can really visualize molecules very easily. Okay, so with the electron microscope, we use the instrument to get 30,000 times magnification. That's a typical number. And to reconstruct an object, many different views must be collected. Now, this is a transmission microscope, electron microscope, which means that the electrons go through the specimen and the image is formed behind the object. And the problem with electron microscopy is that the sample must be very thin because electrons are very readily absorbed by matter. So only something like 20 to 30 nanometers, 200 to 300 nanometers are allowed in thickness. Otherwise, the electrons get stuck. And for the same reason, the column through which the electrons pass are at very high vacuum because electrons are also get stuck in air. And the electrons also strongly damage the molecules, and there is a need for low dose. The typical doses that are used are between 10 and 20 electrons per square angstrom. And that has the effect that the images are very noisy, which simply means that looking at an individual image of a molecule doesn't make any sense. You don't see anything sharp. We speak of shot noise. The shot noise is created by the random incidence of electrons. And initially, negative staining needed to be used for sample preparation. There was no such thing as cryo-electron microscopy. That only came later. And all the techniques, the mathematical and computational techniques that I have developed were all developed with negative staining. And I'll show you in a moment what that means. Now, just a look at the transmission electron microscope in a bit more detail. There's a high voltage here. And between the cathode and the anode, there is a very short distance. And in this distance, the electrons are accelerated to a very high speed. It's close to the speed of light with 300 kilovolt. And then there's an illumination system made by these magnetic lenses, and the electron beam then goes onto the specimen. The specimen, I'm sorry, this is the illumination system. Here's the objective lens, and the specimen is right here in this plane. And an image, an intermediate image is formed, and that image, it's around here, and that is further magnified by the projector lenses. And in the end, we have an image which used to be on a fluorescent screen, and we could look at it directly. And there was also a plate camera. Meanwhile, there's just an electronic camera here, a digital camera in this plane. So negative staining, the principle is like this. The molecules are essentially in solution, and then a certain concentration of heavy metal salt is added to the solution, and the solution is dried, is air dried. And what what we get is we get molecules with little puddles of stain around them. And so instead of, we don't see the interior of molecules very well, we only see the negative, the stain as it penetrates the crevices. And here is an example of this. So it's not a very good technique of sample preparation, but it gives us high contrast, and at least it keeps the molecule a little bit in its original shape and prevents it from complete collapse. So at the beginning of the molecular type of electron microscopy, there were three different groups at the MRC in Cambridge. The first pioneering effort was done by de Rossi and Klug in 1968, and they looked at the helical arrangement of the bacteriophage. This bacteriophage tail with highly helical order, and then followed by Tony Crowther, who for the first time looked at spherical virus with icosahedral symmetry. And then later on, Richard Henderson and Nigel Unwin looked at a planar crystal and determined structures from that. Now you can see there's something here in common. What is in common is that the molecules are all arranged, they are packed together, they're arranged in a highly symmetric order, and the order itself is being used in the mathematical programs to determine the structure. So essentially you have different kinds of programs to deal with these kinds of structures, and different programs to deal with icosahedral symmetry, and different programs for this planar kind of crystals. So that begs the question, why do we need crystals at all? And why do we need so many different kinds of maths and different kinds of programs? Okay, so my mentor, Walter Hoppe, who was actually an X-ray crystallographer who went into electron microscopy, he already asked the question, why we need crystals? Couldn't we just use the molecules as they are, and they are then now air dried, and they're sitting on the grid, and we simply tilt the grid in many different angles, and so we get all the views that we need, and then in the end we reconstruct the grid in its entirety with the molecules on top, so we can get the reconstructions of these molecules. The problem with this is that we get an accumulated electron exposure with more than 1,000 electrons per angstrom square. Under these conditions, the molecule essentially burns up. So we do get a reconstruction, but the reconstruction becomes completely meaningless. And so I had a different idea on how to approach this. And this idea is the three reconstruction of asymmetrical molecules by single particle techniques. So what does it mean? The structural information comes from images of single, which means unattached molecules that are not crystalline. They exist in many copies in a cell extract, and obviously they are randomly oriented. So they are free to assume all naturally occurring conformations. They're randomly oriented, and a single snapshot may already give us hundreds of particle views. And hundreds is not enough. So we simply continue collect more and more snapshots until we have enough of the angular coverage that we need in order to reconstruct. So this is a very simple idea. However, the problems come in the details, because we don't, to begin with, we don't know the angles, because the molecules have oriented themselves. They have tilted themselves. So we don't need we don't know the angles, so we need to get the angles after the fact. And then obviously the molecules have different structures, because they could change in conformations, they could be in different binding states and so forth. So we need to have some kind of a sophisticated way of sorting them so that we have identical groups. And so there are all kinds of things to be solved: aligning them, sorting them, finding projection angles, reconstructing them in 3D. All these necessitated developing programs, because there were no programs that addressed these issues. And I just only, put some little cartoons here to indicate what kinds of problems needed to be solved: the alignment, sorting, and then finding projection angles. And in order to do this, I developed a program system, a modular program system, which contained many different tools that could all be put together in different ways. So one could build a very large program out of these individual building blocks. So alignment and averaging, proof of concept here, that we're able to align and average individual molecule projections. And then there's the problem of heterogeneity that needed to be addressed, and we developed multivariate statistical analysis. Now, all in all, this is just the beginning of a timeline that stretches to until 2020, and I'm only just showing this to you because all the inventions for which I got the award of a Nobel Prize, they were all contained within the stretch of something like 10 years, between 1975 and 1987. So all these different things were done during that time. And then I have to highlight my hero here, Michael Rademacher, who developed many of the programs that have to do with 3D reconstruction. And through his help, within 3 years, we got the first single particle reconstruction that was published in EMBO Journal in 1987. It's a reconstruction of the 50S ribosomal subunit from E. coli. And this here is just shown with different density threshold levels. And then came the very big breakthrough through the introduction of the cryo techniques. So the molecules could be embedded in vitreous ice, and the efforts were mainly through Jacques Dubochet, who came out with this technique in 1981. And the breakthrough really came from realizing that you cannot simply plunge the grid into liquid nitrogen. You have to plunge it into liquid ethane or propane. Because if you plunge it in liquid nitrogen, you get air bubbles that are formed at the interface between the grid and the cryogen. And these air bubbles prevent the heat from being transferred rapidly. So here with propane or ethane on liquid nitrogen temperature, you get very fast heat transfer. And under these conditions, we prevent the formation of crystalline ice. We get vitreous ice. Vitreous ice has the same consistency as water, and or you know, sort of a glass form of water. And under these conditions, the molecules are are essentially preserved in their native state. And here is are these plunging devices. This is a commercial plunging device, cost in the range of $50,000, and one can build one by hand for $200. But the difference is that this is climatized and has a way of operating the blotting and the plunging under computer control. So the idea is that we have the grid on the tweezers, and we have a droplet of the liquid where the molecules are in, is put on the grid, and then the excess is blotted away so that we get a very thin layer. And then the grid is plunged into liquid ethane. So these, for instance, this is the image that you get from ribosomes. Now it looks so faint because of the salt and pepper effect that we get from the shot noise, and it's also so faint because of a defect of the lens of the microscope, the contrast transfer function. It has the effect that we have very, very low contrast of the particle itself. It doesn't stand out from the background very well. But nevertheless, with these techniques now, the technique of single particle reconstruction and cryo combined, we got the first beautifully preserved reconstructions of molecules, but still at rather low resolution. The resolution range is in a range of between 20 and 30 angstroms. And all these molecules had never been seen before because they could not be crystallized. And this is the first very detailed picture of the E. coli ribosome, and then many different forms of hemocyanin, calcium release channel. This is the kind of microscope that used to be there that is already very specialized for cryo techniques. And the first really beautiful microscope of this kind was the Polara, so-called Polara microscope. And we got one while I was still in Albany in the year 2000. So in general, we now sort of have a pipeline in biological cryo-EM. We start from biochemical preparation, cryo-EM sample preparation, imaging, data collection, image processing, reconstruction, structural analysis. And then if we get high enough resolution, we can trace the atomic structure from that. So we can, if we get in the range of 3.5 to 3 angstrom resolution, then we can already do that. Now, along the way, there was the invention of of maximum likelihood methods of classification, and it turned out that by using maximum likelihood classification, we are able to actually determine several structures all at once. So they can be in a mix in the image, and we can extract all the different 3D structures from the same sample. So I'm calling this story in a sample. So it's really fantastic. We can get several structures out of this. And for instance, if you have a molecular machine and it's doing its whatever the machine is doing, then we get several different states of that machine all in the same sample, and we can extract all of them separately. And then another very big revolution occurred, which was in 2012. Very highly sensitive cameras were were developed by 3 different manufacturers, and they are much better than film. And this is essentially the MTF, modulation transfer function. And you can see they're remarkably better than film. So with these cameras, it is possible to actually see every single electron that impacts. And here's a field of ribosome recorded on such a direct electron detection camera. So that really brought a breakthrough. The first such high resolution structures that got in an atomic resolution range came out at UCSF, and it was the first reconstruction of an ion channel, where the ion channel was surrounded by a lipid, by some kind of a lipid bubble, which fakes the membrane for the protein. And in our group, with a focus on ribosomes, we got elongation factor G in a mutant form, and you can see here that we get close to atomic resolution, and that 4 different structures are extracted all at the same time from the same sample. And they give us an idea about particular different states of the interaction of the elongation factor G with the ribosome. And then the most spectacular result that we got in our lab was the determination of the structure of the T. cruzi ribosome at 2.5 angstrom resolution. And it was really fantastic to see the base pairing in detail. We see individual ion, magnesium ion, zinc ion. We see even the position of water molecules. So I'm just going to give you a flavor of this reconstruction here. I'm spinning it around. It's the small and the large subunit is on top. And now we're showing the different local resolutions. The resolution is different at different parts of the molecule. The interior is the most stable part and it has the highest resolution. And the periphery, obviously, can be floppy. Okay, so now before I showed you this Polara microscope, nowadays the microscopes have become huge and completely self-contained. And this is the Thermo Fisher Titan Krios at 300 kV, and you can see the sheer dimensions of that. And it requires an, essentially different kinds of buildings around it. And this is the inside of it. You can see there's a heavy mixture of electron-optical components, and computer components. Here's a close look at the positions close to the objective lens. So when all these development occurred, and there were not many microscopes around. There was a lot of interest in New York of people collaborating with me on all kinds of molecules, and these are many were ion channels and receptors. Here's the AMPAS receptor that was done by one of my students in collaboration with another faculty member. So from the ribosome research, I branched out to all kinds of other collaborations. And one of the most important ones was the ryanodine receptor. And this is a collaboration with two other groups at the Columbia Medical Center. And these two groups, Hendrickson and Marks, had collaborated for 9 years trying to get an X-ray structure. And they got stuck at 7 angstrom resolution. And then within a year of collaborating with us, we were already at 4.5 angstrom resolution, and now we are at close to atomic resolution. Cystic fibrosis is one of the molecules of very high public health interest, and we've been working on the mutant that is important for the disease process. So, and meanwhile, the resolution has reached something like 1.5 angstrom resolution, even 1.2 in one case. So you can see that sort of everything rapidly develops. The instruments become more sophisticated and specialized. And this here I show you because we can now see the signatures of individual amino acids. When we reach this kind of resolution, we don't need to know the sequence anymore. We can find it simply by going through the 3D structure. And then this here is really very important. Just in time, the single particle cryo-EM arrived just in time for us to deal with the new pandemics. And we have MERS, Ebola, Zika, dengue, SARS, and now the COVID virus. And in all these cases, after 2012, when we reached the sufficient resolution, in all these cases, the cryo-EM has been instrumental in making the initial discoveries as well as helping in the development of drugs. And it has been a factor in the development of the recent vaccinations. We just show you. And so last year, just 2 weeks after receiving the sequence, this structure of the spike protein was determined by McClellan at Arizona State. And we are at Columbia, we are looking at the visualization of neutralizing antibodies against multiple epitopes. And this is something that just appeared half a year ago. So the number of released structures in the PDB per year is pretty much leveling off for X-ray. NMR, it slightly went down. And you can see that for cryo-EM, it's rapidly moving up now. So this is the rate per year. So the rate is going up. So in conclusion, Single particle cryo-EM opens a new era in structural biology. There's no need for crystals. Very small sample quantities are needed. Resolution is in the 3.3 to 4 angstrom range, now routinely available. And the best resolution so far is 1.5 angstroms. Multiple structures can be retrieved from the same sample, which can give us clues on function. Molecules are enclosed to native conditions, and solving structures of membrane proteins is much easier than with X-ray crystallography. And in effect, we now get a huge expansion of structural database relevant for molecular medicine. And I just have a few slides that might be interesting because it's such a fabulous event arriving in Stockholm and being greeted like this. Here we're sitting around for a rehearsal. You see, everybody has very informal clothes. It's a rehearsal of the ceremony. And here I am at the museum. The Nobel Museum asks every Nobel laureate to bring something that is somehow reminiscent of the beginning of the whole discovery or invention. And so I brought with me a box of mounted transparencies, where each transparency was for a slice of the structure. So this was an old way of representing a 3D structure on a light box. Next to me is Jacques Duboucher, who presents his initial plunger. And on the other side is Richard Henderson, who presents his atomic model of bacteriorhodopsin. Okay, here we are with Alfred Nobel. Here I am with my wife, Carol, who's been incredibly supportive all this time. And here we are. This is in the year after I got my award. We are standing on top of the building that houses our lab. Thanks very much for your attention.
Oh, this is incredible. Thanks, Dr. Frank. Yeah, just really amazing how applicable it is to especially today's pandemic. So we want to open up the floor for questions. And I think the way that I can do this is if people can raise their hands, I can unmute them, so that way we have an organized question platform. So anyone who wants to ask a question, okay, I see that Gilbert raised his hand. Or actually, Dr. Tang, I'll unmute you first.
Dr. Frank, thank you for this very inspiring talk to our students. Most of our students are PhD students and they're in the early part of their career. And look at your history of invention start from 1970s to now. That's amazing long time work, persistent. Congratulations for your winning Nobel Prize. I'm a food engineer. So we don't, what we do is we develop technology to control pathogens. You mentioned about E. coli in the food systems. So one area of study we started about 10 years ago is trying to inactivate Salmonella, and those are very tough, Listeria, Salmonella, very tough bacteria that survive in a dry environment. And our suspicion is that the ribosomes are the key weak link in the cell that can inactivate during the thermal inactivation. When they rehydrate, they do not reproduce anymore. So, but we are puzzled about what would be the structure of the ribosomes in the dehydrate stage. You show this nature kind of form, use a cryo-EM to show the natural form of the ribosomes. But what happens if the ribosome is dehydrated? And our suspicion is that dehydration removes the water and make this structure of ribosome very rigid. And so that when you increase the temperature, it's very difficult to inactivate, do the irreversible denature. So I'd like to hear your opinion on this. And secondly, how we can collaborate, if possible, to review the structure in a low environment. This is a major problem with food industry, where you see the baby formulas contaminated with salmonella. When they rehydrate, get food disease, foodborne disease, but also in many other spices, in flowers. So those is a major problem with food industry that we started the research about only 10 years ago. We still start to understand why and how, but fundamentally, I like to, this is your talk is very interesting to me, and I really like to hear your opinion about what happens to the bacteria when they're heated by temperature, heated in its treatment, and what happens to the cells.
I'm really I'm a specialist in structural biology, and I know quite a bit about ribosomes, but only in their native environment, and not if they are mistreated like this. We obviously we don't have an insight on the structure of ribosomes that are dehydrated, because that's not possible with the techniques that we have. We can only imagine what will happen. Now, the ribosomes are very rigid because of the high RNA contents, so that they don't get denatured that quickly. But obviously, there's the protein contents, and they can become denatured. And I really don't know. We know a lot about from comparison between thermophilic bacteria with the normal bacteria. We can see how they essentially maintain function in very adverse environments, and so a lot of insight one can get from there. But I'm not I don't know anything about studies of dehydrated organisms. Tardigrades are probably a very good place to look, because they are just amazing in keeping themselves alive.
Yeah. So just for your information, for example, if the bacterial cell is in a high moisture environment, they can be killed like 60 degrees C in a couple of minutes. But if they are dehydrated, it takes about 100 minutes, the same temperature, 100 minutes, about several orders of magnitude. So that makes this a very interesting for us, but also it's very tough for food industry to deal with. It's very tough to inactivate when they're in the dehydrated stage. So that's very interesting, topic that we started about 10 years ago. But thank you very much.
Yeah.
I have another question in the chat from Jose. In 1990s, after coming down from string theory and 10 dimension, I found along the way unfolding dash unfolding in 2 dimensions and some math properties in knot theory. Have you considered connecting this protein folding unfolding with knot theory? I don't know if that question was clear.
No,
Jose, would you like to? unmute yourself.
I don't know much about protein folding and unfolding. I'm wondering if you know about the topoisomerase that we have in our cells. It's very interesting because if they're actually pseudonodes form or actual knots, they're broken down by topoisomerase. So the strands are disconnected and reconnected in order to remove knots. And then we have this process going on all the time. The topoisomerases, they're marching up and down the DNA to look for spots that need to be resolved.
Great. I hope that answered Jose's question, I think. And so we have another question. Basaraj, I see you raising your hand. Okay, you should be able to unmute now.
Thank you, Dr. Frank. I have a general question one. So from the first slides, I got one question that incidentally in today morning, I got one article from Quant magazine titling "What is Life?" So it's explaining that how the diversity of life from snowflakes, RBCs to tardigrades makes it difficult to define it. So when you looked into the ribosomes ultrastructure, how you would like to define it? Like, so how would you like to define the life in the perspective of ribosomes?
Well, I don't know. I mean, from a very pragmatic way, I would say life is if you have ribosomes, okay? Because it's like an exhaustive definition. Simply looking around, I would say that if an organism doesn't have a ribosome, it cannot live. But that definition doesn't help anybody, obviously. So I mean, knowing anything about ribosomes doesn't give me a particular head up in defining life. I mean, there are many definitions of life that imply the ability to replicate, sustaining yourself in adverse environments and so forth. There's a whole list that people come up with. There was a very interesting article just recently about this, about the difficulties in defining life, because whatever definition you come up with, you always run into exceptions.
So just a thought. So why don't we include time as a factor while defining the lives?
Why don't we include what?
So why don't we include time as a factor while defining the life? Because
Time?
as a factor, yeah.
Time, okay. All right, please go ahead.
Yeah.
I don't, yeah, time, I think time we would include everywhere, wherever we want to talk about something. Yeah.
That's what I thought, because most of the definitions just go with the ultrastructure or life forms or living entities that we found in terms of molecules or nuclei or something. But we never thought in terms of the time those molecules or ultrastructure we find. So we don't include time as a factor in defining those things that I found. And yeah, just to have a thought from you. But thank you. Yeah.
All right. Do we have any further questions? We're scheduled until 10 o'clock. I'll give people a few more seconds to ask a question. So Dr. Frank, I had a quick question about the Nobel Prize in particular. So from your presentation, I didn't really grasp that you had collaborated with your co-recipients so much as you all contributed to the same technology.
Well, we didn't collaborate. We did not collaborate.
Mm.
But the technologies merged. They merged together.
That's a beautiful thing, really.
Mm-hmm.
All right, I see one more hand from Dr. Tang.
Well, Dr. Frank, any advice for our students who have ambition to change the world, and of course have to start from step by step? What's your advice for our students? A young scientist.
Yeah, I'm being asked about this a lot. This is really, from my own experience, what I would advise is if you have really a very good idea and you're convinced about this and it checks out, then don't let yourself be talked out of it. And so sometimes, listening to a mentor can be inspiring in both ways, and you can develop something completely different. You can get inspired to do essentially the opposite of what you're advised. So this is one thing, and the other one is what I keep getting back to is peripheral vision. Look at things that are sort of coming up on the wayside. So if you just very strictly follow one plan, you might overlook opportunities that come along. Very often there are complete accidents, accidental discoveries, and many accidents on the way. So my entire career is really an assortment of directions and misdirections in all different ways. So it was not a straight path at all, and so that's essentially my advice. Peripheral vision, that's the important thing.
Thank you. Jack, do you want to give a chance to Geshem and your chair to thank Frank, Dr. Frank?
Yeah, sure. So Dr. Perez Garcia, are you, let me see, are you available to be unmuted? Oops.
Thank you very much for this inspiring talk. Our students, really, this is all made for our students, and we want them to continue leading the way. We're in the back. Really, we hope that they are. And again, please, if you ever come to Washington State, make some time to visit the Palouse. We have a beautiful region, and we'll be very happy to host you around. And again, we're very pleased, and we learn a lot. But overall, I think for our students in this is the inspiration that they can get wherever they want to go. So thank you very much for being that inspiration for our students today.
Thank you.
Yeah, thank you, Dr. Frank, and thank you everyone for attending today. That just about wraps up our hour. I hope everyone has enjoyed this, and that you all have a great rest of your day.
Okay, thank you.
Thank you.
Stay healthy and stay safe.
Thank you.